now let's go ahead and talk about how to deal with confounders in observational studies. So we've been talking about these observational studies and how correlation doesn't imply causation, and there can be these confounders mixing up our results. So the question is, what can we do to help make our study better? And I'm going to start off with the lighters and lung cancer example. So we've seen that observational studies show that people who carry lighters have a higher rate of lung cancer. And with any observational study, there are two possibilities. The first one is that the treatment causes the response, so that the lighters cause the lung cancer. And if this is true, then there will be a causal link explaining how or why that is the case. The more likely option is number two, that the lighters do not cause lung cancer. And instead, there is a confounder or a third factor that is messing with our experiment, making it look like the treatment causes the response. So, as we dive into this problem, we're going to really quickly just identify three things as either confounders, causal links, or neither. So, we're going to get more practice with the diagrams. And it says to create your own diagram to decide how to classify these things. So, the first one is genetics. So I'm going to go ahead and draw my diagram. Lighters is my treatment. Lung cancer is my response. And then we want to know what is genetics. So based on the information given, it says some people have certain genetics that make them more likely to get lung cancer than others. So right away, I can see genetics can lead to lung cancer, but here, no mention of lighters. So this is definitely an example of something that is neither a confounder or a causal link, because it's not connected to our treatment. It's just kind of a random fact about the response. The next one is smoking cigarettes. So we know for sure that smoking cigarettes does cause lung cancer. It's one of the known causes. So I have lighters as my treatment, lung cancer as my response, and then smoking cigarettes in my box. And like I said, it says smoking cigarettes is a known cause of lung cancer. So I'm going to draw the arrow from smoking cigarettes to lung cancer. And then it says people who smoke cigarettes are also more likely to carry lighters. So with this example, this is a classic example used to teach confounders. And smoking cigarettes is usually the obvious one, the obvious confounder. So after we look at our very last option here, we're going to look at how can we confirm that smoking cigarettes actually is a confounder. So then the last example, just to go ahead and finish this up, is lighter fluid. So I have lighters as my treatment, lung cancer as my response, and then lighter fluid in the middle. And it says perhaps inhaling the fumes from lighters can cause lung damage leading to cancer. So this is saying, okay, we have these lighters, we get the lighter fluid, we inhale it, and then that could get into our lungs and cause cancer. So this one, slightly different than the confounder, is saying, what about the lighters causes the lung cancer? It's the lighter fluid. So lighter fluid is something about the lighters, whereas smoking cigarettes is a third factor. So this is the classic causal link. It explains how or why. So we see three examples here, neither confounder and causal link. And I said we're going to focus on that confounder. We're going to focus on this smoking cigarettes. It says, how can we determine whether it's the lighters or the smoking that's the true cause of lung cancer? So we know this is the case, but back when they didn't, how did they determine that it really was the cigarettes and not the lighters? So there's a method called stratification that helps us deal with confounders. It's one of the most common ones. There's another method called regression that we'll get into way later. But let's look at stratification for now. So stratification is actually very similar to blocking. In randomized controlled experiments, if we have small sample sizes, we can block or divide our subjects into subgroups based on certain factors. And stratification is essentially the same thing for observational studies. So it says stratification involves breaking the treatment and control groups into smaller, more homogeneous subgroups where the confounding factor is the same. So in this case, we have our potential confounder, which is smoking. So we want to check to see if smoking is a confounder. And we can do this through stratification. One way to deal with that. So what we did was actually divide our subjects into smaller, more homogeneous subgroups. We looked at non-smokers, moderate smokers, and heavy smokers. And basically, instead of making one overall comparison, we want to make 3 separate comparisons. We're kind of trying to compare apples to apples within each group. So let's look at this diagram. It says, in this case, researchers saw no difference in lung cancer rates between those who carried lighters and those who didn't within each group. So I think the diagram explains this really well. We have the start of the study, right? We have everyone in the study right here. And we saw that the treatment group, right, the group that carried the lighters, actually had a higher rate of lung cancer than the control group who had no lighters. So this balance scale is kind of on a scale of our response, right? We're looking at lung cancer rate. And so we don't want to say, because this was an observational study, we don't want to say, oh, okay, that means the lighters caused the lung cancer. Instead, we thought smoking could be a confounder. So let's go ahead and divide our subjects into subgroups based on smoking. So we first looked at all of the non-smokers. So we looked at the non-smokers who were in the treatment and control groups, and we actually saw that it didn't matter for the non-smokers, whether they carried lighters or whether they didn't, they had a relatively low rate of lung cancer. Again, if this is my scale of the lung cancer rate, we can see that that difference went away when we just looked at non-smokers. Non-smokers in both the treatment and control group had a pretty low rate of lung cancer. So this is already evidence that maybe it is the smoking that's driving that difference. So then we decided to look at the moderate smokers next. So we have the moderate smokers, and we saw the same thing. That difference went away. The treatment and control group both had kind of a moderate rate of lung cancer. It didn't actually matter if they carried lighters or not. What mattered was the moderate smokers had a higher rate of lung cancer than the non-smokers. And then lastly, the heavy smokers, we confirmed our suspicion. The heavy smokers, regardless of if they carried lighters or not, had the highest rate of lung cancer. So the increase in lung cancer rate was actually due to the smoking, not the lighters. If we look at the treatment and control group in each group, they're balanced, they're the same. So here we confirmed that smoking was the confounder. So I think this diagram helps see it visually. Up here in writing, it says, in this case, the researchers saw no difference within each group. The heavy smoker group had the highest lung cancer rates, and the rates between those who carried lighters and those who didn't were actually the same. So we saw that the smoking caused the lung cancer and not the lighters. So here we can see observational studies with confounders can be very dangerous because we could actually come up with the wrong conclusion if we don't think about confounders and then adjust for them by stratification. So at the end here, it says with observational studies, you actually need a much bigger sample size. And you'll see this a lot. Sometimes you'll have tens of thousands of people in observational studies, because with those, we know that we're eventually going to have to probably adjust for confounders and do something like stratification. And every time you stratify, your group gets smaller and smaller. So the last thing I want to talk about is how would you stratify to account for a second confounding variable? Because you can have, most of the time you have multiple confounders in these observational studies. So let's go ahead and say that we thought maybe income was a second variable that could be a confounder. So we would stratify basically first based on smoking and then based on income. So we have our treatment group, right? The group that carried the lighters. And then we have our control group, the group that had no lighters. And with stratification, we want to make multiple comparisons. So we said, okay, we divided the treatment group into non-smokers, moderate smokers, and heavy smokers. And then we did the same for the control group. And then we made sure to compare apples to apples. We want to compare the non-smokers in the treatment to the non-smokers in the control. We want to compare the moderate smokers in the treatment to the moderate smokers in the control, and same with the heavy. So if I wanted to add on income as a second variable to stratify with, I would actually have to make even smaller groups. I'd first take, so let's just look at the non-smokers in both groups. I'd have to divide them into, I don't know, I could do high income, medium income, and low income. So that's taking those non-smokers, dividing them into 3 groups. In the treatment group, I'd have to do the same in the control group. Take the non-smokers and look at them, the ones that had high incomes, medium incomes, and low incomes. And then you can see, I'm kind of making a tree here, that it gets complicated really fast. So now I have to make even more comparisons. I'm going to compare the non-smokers with high income in the treatment group to the non-smokers with high income in the control group. Then I have to do that for the non-smokers with medium income, et cetera. And you can see how it can get super complicated. I also have to divide the moderate and heavy smokers into those 3 groups based on income as well. So you definitely need a big sample size so that you don't run out of people when you're stratifying. So again, this just shows this complicated stratification, especially with more than one variable, shows that these observational studies aren't ideal, and that whenever possible, it's best to do a randomized controlled experiment.